Let's jump straight into number 10, the historic collision of measles and cattle. About 2.5 millennia ago, an event unfolded on the vast Eurasian grasslands that would dramatically redefine human history in ways none of the people living it could possibly have comprehended. Humanity had begun domesticating cattle on a grand scale, bringing these immense creatures into painfully close proximity with human settlements, a dangerous new intimacy for our species. And deep within those cattle, a lethal virus known as rinderpest circulated, a disease so devastating it could obliterate entire herds in mere weeks. Then, genetic analysis published in the year 2020 by scientists at the Robert Koch Institute definitively pinpointed the precise moment rinderpest made its terrifying leap. It wasn't just a jump, it dramatically hurdled the species barrier, invading humans, and from that transformation, something entirely new and far more insidious emerged. That something was measles. The virus didn't merely adapt, it underwent a profound metamorphosis. Measles developed an airborne transmission route so frighteningly efficient that a single infected individual could readily pass it on to between 12 and 18 other people within an unvaccinated population. No other human virus in recorded history approaches this staggering rate of transmission. The R0 value of measles makes it the most contagious virus humanity has ever confronted. It's far more contagious than influenza, more contagious than Ebola, even more contagious than the coronaviruses that have so dramatically reshaped our modern world. Every single cough, every exhalation, every shared breath in any enclosed space holds the terrifying potential to infect anyone who hasn't previously been exposed. What makes measles uniquely disturbing, however, is the catastrophic impact it inflicts directly on the human immune system itself. Groundbreaking research published in the journal Science in 2019 shockingly revealed that measles doesn't just cause a temporary illness. More profoundly, it systematically erases immune memory. The virus devastatingly wipes out a staggering 11 to 73% of the protective antibodies that shield you from other diseases you've already survived. After a measles infection, your body essentially forgets how to battle illnesses it had already learned to defeat. Chickenpox, various flu strains, bacterial infections you had acquired immunity to over decades, all of it is gone. Your immune system is brutally reset to a state shockingly similar to that of a newborn infant, leaving you vulnerable once more to pathogens your body had victoriously vanquished years before. The chilling implications of this immune erasure resonate through every single epidemic that has followed. When measles arrived in the Americas with European colonizers, it didn't just kill the people it directly infected. It left the survivors completely stripped of their immune memory, utterly defenseless against every other disease relentlessly sweeping through their vulnerable populations. Entire magnificent civilizations crumbled, not from the impact of a single virus, but from the horrifying cascade of subsequent infections that measles so brutally made possible. This virus, which explosively emerged from a cow 2,500 years ago, has sculpted the genetic and demographic structure of modern human species with a deeper and more profound impact than almost any other event in all of recorded history. Measles, despite relentless vaccination campaigns and decades of devoted public health efforts, is still here. The virus stubbornly persists in pockets across every continent. Alarming outbreaks in the year 2024 have already reached levels not witnessed in well over a decade, with the World Health Organization grimly recording a 79% surge in cases across Europe alone. The virus patiently awaits its opportunity in every unvaccinated population, every single region where immunization coverage precariously drops below the 95% threshold critically required to halt its transmission. And every single time it resurges, it brings with it the exact same immune-erasing legacy it has carried since those Bronze Age grasslands where it was terrifyingly born. Every modern measles outbreak isn't merely a public health failure, it's the chilling reawakening of an ancient event that began 2.5 millennia ago, when a cattle virus crossed dramatically into human bodies and discovered it profoundly preferred to stay. Moving on to number 9, papillomaviruses and their ancient roots in the deep mammalian past. Long before humans even existed, long before primates evolved, long before mammals developed into anything remotely resembling their modern forms, a specific family of viruses had already firmly established its reign within the flesh of ancient creatures. Papillomaviruses have been inexorably co-evolving with mammals for hundreds of millions of years. Phylogenetic analysis, meticulously published in the journal PLOS Pathogens in 2013, traced the unbroken lineage of these viruses all the way back to the Jurassic period, strongly suggesting they were actively infecting the ancestors of modern mammals even as dinosaurs still roamed the Earth. These truly are not recent arrivals. These viral passengers boarded our evolutionary vessel long before mammals ever developed fingers, before primates grew tails, even before anything remotely resembling humanity walked the earth. The virus itself reproduces through a unique mechanism that directly embeds itself within the skin cells and mucous membranes of its host. There are more than 200 distinct types of human papillomavirus identified so far, and researchers estimate that a staggering 80% of sexually active adults will encounter at least one strain during their lifetime. Most of these infections will quietly resolve themselves. The host often remains completely unaware they ever harbored the virus. Yet some strains refuse to relinquish their hold. They integrate themselves deep within the genetic machinery of the infected cells, beginning an insidious process of rewriting the fundamental instructions that these cells have diligently followed for hundreds of millions of years. What makes papillomaviruses particularly unsettling is the sheer, overwhelming scope of disease they inflict upon modern human populations. Cervical cancer, astonishingly the fourth most common cancer among women globally, is almost entirely a direct consequence of persistent HPV infection. The World Health Organization estimates that HPV is responsible for approximately 690,000 cancer cases each and every year, a grim tally that includes cancers of the throat, the anus, the penis, and the vulva. This ancient virus, which first emerged during the age of dinosaurs, remains the single largest viral cause of cancer in the human species, responsible for more malignancies than all other viruses combined. The very persistence of this virus defies our intuitive understanding of mathematics and survival. Papillomaviruses have not only endured, but also thrived through every single mass extinction event that has scourged the Earth since their primordial emergence. The asteroid impact that famously wiped out the dinosaurs 66 million years ago did not eradicate them. The cataclysmic ice ages that repeatedly reshaped our planet did not eliminate them. Even the rise and fall, the emergence and eventual extinction of entire orders of mammals did not extinguish their relentless march. These viruses simply exhibited an unparalleled adaptability, jumping effortlessly from one host lineage to the next, precisely tailoring themselves to each new species, thereby carrying their genetic payload forward across timescales that reduce the entirety of human civilization to a mere rounding error. The truly disturbing implication of this astounding persistence lies in what it reveals about the fundamental relationship between viruses and complex life itself. Papillomaviruses may not be invaders of the mammalian body in the conventional sense. Instead, they might be permanent residents, meticulously co-evolved right alongside us. Their presence is so ancient, so utterly and thoroughly integrated into the very fabric of mammalian biology, that it becomes almost impossible to imagine our species existing without them. Every wart, every suspicious cervical lesion, every throat cancer driven by these persistent viruses is a modern echo of an infection that took root long before mammals even comprehended what it meant to be warm-blooded. We did not evolve to be free of these specific viruses. On the contrary, we evolved in tandem with them, and they, in turn, evolved to become an integral part of us. Neither of our species has ever truly existed without the other. The human species, like every mammalian species before us, will eventually face extinction. And when that inevitable moment arrives, papillomaviruses will simply transition, jumping to whatever new creatures remain, adapting to new hosts with the same unwavering efficiency they have displayed for hundreds of millions of years. They will outlive us. They have, in fact, already outlived almost everything else. Number 8. Influenza A and the Ancient Reservoir. Deep within the primordial wetlands of the prehistoric world, thousands of years before humans even conceived of building cities, a cunning virus was already circulating tirelessly through the bodies of countless waterfowl. Ducks, geese, and various wild aquatic birds silently harbored Influenza A within their intestinal tracts, continuously shedding it into the very waters where they instinctively fed and bred. Groundbreaking genetic analysis of influenza lineages strongly suggests that this virus has been actively circulating within avian populations for at least 4,000 years, and quite possibly for an even more extended period. The birds themselves were rarely, if ever, harmed. They had, through eons of coevolution, developed a remarkable tolerance for the virus's presence, much like a thriving forest tolerates the essential fungi that decompose its fallen dead. But these birds naturally shared their environments with every other species that came to quench their thirst from the same shared waters. And the virus was perpetually poised, patiently awaiting its opportune moment to cross the species barrier. The very structural composition of Influenza A grants it a unique and terrifying capability for leaping between different species. The virus carries two crucial surface proteins known as hemagglutinin and neuraminidase, which exist in dozens of distinct variations. When two different strains of the virus manage to infect the same host cell simultaneously, their genetic segments can undergo a dramatic reshuffling, creating entirely novel and unprecedented combinations that no immune system has ever encountered before. This process, known as antigenic shift, is the primary mechanism that has driven every major influenza pandemic in recorded history. This rapid genetic recombination, known as antigenic shift, is the very engine behind every influenza pandemic etched into recorded history. It was this molecular dance that unleashed the devastating 1918 Spanish flu, a plague that claimed an estimated 50 million lives. The same mechanism birthed the 1957 Asian flu, the 1968 Hong Kong flu, and the more recent 2009 H1N1 pandemic. What truly elevates Influenza A to a terrifying level is its inescapable permanence. We cannot eradicate it. This insidious virus thrives in the wild bird populations across every single continent, carried on invisible currents along migratory flyways that literally crisscross our entire planet. Each year, countless birds become winged carriers, transporting novel variants thousands of miles, scattering them into new ecosystems and exposing them to unsuspecting mammalian hosts. Pigs, in particular, become unwitting crucibles in this viral alchemy. Their respiratory systems are uniquely vulnerable, capable of simultaneously hosting both avian and human influenza strains. This dual infection creates a biological mixing pot, allowing the virus to reshuffle its genetic components within a single animal, emerging as a chimeric threat. The result? A new pathogen against which neither its avian forebears nor its human targets possess any inherent defense. History screams a clear warning, revealing how frequently this monstrous process has impacted humanity. Researchers meticulously sifting through ancient DNA and age-old accounts have traced probable influenza pandemics stretching back millennia. Ancient Greek medical texts, meticulously kept Chinese imperial records, and somber medieval chronicles all paint chilling pictures of outbreaks mirroring influenza's signature spread. A rapid, relentless tide of respiratory illness, leaving a trail of high mortality among the elderly and the youngest. The virus has been a silent executioner for as long as we've documented our own diseases, and it stalked our ancestors long before the first word was ever written. The truly chilling aspect of Influenza A isn't just its past, but the cold, hard inevitability of its future. The virus isn't gone, it is ceaselessly circulating in billions of wild birds, endlessly reshuffling its genetic hand every single time two strains manage to meet within a single host. Epidemiologists don't waste time asking if the next influenza pandemic will occur. Their vital questions are when and how many souls will perish before it can finally be brought under control. The primal virus that first emerged in the ancient waterfowl of a prehistoric world is far from finished with us. It has simply been biding its time, waiting for the precise genetic combination that will be devastatingly effective at sweeping through our modern human population. And with every passing year, that terrifying combination inches statistically closer. Now we turn to number 7, smallpox and its frozen archive of terror. The earliest irrefutable physical evidence of smallpox scars the preserved body of Pharaoh Ramses V, who succumbed in the year 1157 BCE. His mummified remains, carefully examined in the 20th century, display the unambiguous pustular lesions characteristic of a variola infection. Yet, cutting-edge genetic analysis of viral DNA recovered from other ancient remains hints at a far older, darker truth. The virus predates the Egyptian record by many centuries. A groundbreaking study published in the prestigious journal Science in the year 2020 meticulously analyzed smallpox DNA extracted from Viking Age skeletons. The shocking discovery? The virus had been actively circulating in human populations for at least 1,400 years before the Egyptian evidence surfaced. And phylogenetic modeling, pushing the boundaries of our understanding, suggests origins that could stretch back tens of thousands of years into the deepest recesses of human prehistory. Smallpox, a name that still evokes a shudder, claimed approximately 300 million lives in the 20th century alone. This virus inflicted a disease of extraordinary brutality. It began with fever, followed by a hideous rash, then pustules that erupted across the entire body, often leaving survivors with permanent blindness. Mortality rates swung wildly from a terrifying 30% to a staggering 90%, depending on the specific strain. It stands as one of the most lethal infectious diseases in all of human history, a silent sculptor of destiny that shaped the outcomes of wars, orchestrated the downfall of empires, and irrevocably altered the demographic tapestry of entire continents. When smallpox was unwittingly carried to the Americas by European colonizers, it unleashed an unimaginable horror, wiping out an estimated 90% of the indigenous population across two vast continents within a mere century. What makes the smallpox story particularly disturbing, despite its proclaimed defeat, is the very nature of its official eradication. In 1980, the World Health Organization triumphantly declared smallpox eliminated from the wild. The last naturally occurring case, a grim milestone, had been recorded in Somalia in 1977. Global vaccination campaigns had seemingly driven the virus to utter extinction within human populations. But the truth is, the virus was not actually extinct. Two laboratory samples were deliberately preserved. One of those samples was meticulously stored at the Centers for Disease Control in Atlanta, while the other was held at the Vector Institute in Russia. This decision to preserve these smallpox samples was a deliberate, calculated move, justified by the supposed necessity for future research. Yet, their continued existence means that smallpox, the ancient killer, isn't truly gone. It merely sits, frozen, incarcerated in freezers, patiently waiting. But the truly unsettling truth stretches far beyond these officially sanctioned laboratory samples. For millennia, smallpox victims were buried in the frozen ground across the vast northern latitudes. In the deep permafrost of Siberia, Alaska, Scandinavia, and northern Canada lie the bodies of countless individuals who succumbed to smallpox during outbreaks that span centuries and even millennia. Now, as global temperatures climb and the permafrost begins its unprecedented thaw, these ancient graves are being exposed to the surface, a sight never before witnessed in recorded history. In 2004, a team of researchers exploring a 300-year-old burial site in Siberia unearthed tissue samples containing fragments of variola DNA. This chilling discovery unequivocally proved that smallpox genetic material can persist, viable and intact, within frozen corpses for hundreds of years. The critical question, whether any of that ancient viral material remains capable of causing infection, is one that researchers have, understandably, been reluctant to definitively answer. The mathematics of this looming threat are starkly clear. The Arctic alone encompasses an estimated 15 million square kilometers of permafrost, and this permafrost is melting at a rate far exceeding any climate model predictions from even a decade ago. Hidden within its icy grasp lie the bodies of every person who died from every conceivable disease over thousands of years of human habitation in the north. Smallpox, tragically, is just one among these silent, frozen pathogens. Each year, more of this vast, ancient graveyard emerges into our modern world, and with every passing year, the chilling probability escalates that something buried within that melting ice will inevitably find its way back into a living human body. The eradication of smallpox was hailed as one of the crowning triumphs of modern medicine, a monumental victory. But it was, in reality, a temporary truce. The virus persists, locked away in freezers, entombed in ancient graves, and still encoded within the genetic material of countless corpses preserved in ground that is no longer permanent. The elimination of smallpox from the living world did not, in fact, eliminate the virus from our planet. It merely shifted it into storage, and history teaches us that storage has never offered an absolute, unbreakable guarantee against anything. This brings us to number 6, Hepatitis B and its astonishing Bronze Age passenger. In 2018, a team of researchers meticulously extracting ancient DNA from human remains scattered across Europe and Asia unveiled a discovery that fundamentally reshaped our understanding of viral history. They had successfully recovered intact genetic sequences of the hepatitis B virus from human skeletons dating back an incredible 7,000 years. This ancient virus, they revealed, had been actively circulating within prehistoric human populations throughout the Neolithic and Bronze Age periods, silently spreading across continents, evolving into distinct regional lineages, and infecting humans long before any written record could possibly document its existence. Subsequent studies pushed this timeline even deeper into the past. Research published in 2021 identified hepatitis B lineages that had diverged from a common ancestor more than 20,000 years ago. Today, hepatitis B continues its relentless assault, currently infecting approximately 296 million people worldwide, according to the World Health Organization. This insidious virus causes chronic liver inflammation, cirrhosis, and ultimately liver cancer, tragically claiming an estimated 820,000 lives every single year. Despite the widespread availability of an effective vaccine since the 1980s, the hepatitis B virus remains one of the most pervasive infectious agents on Earth, tirelessly transmitted through blood, sexual contact, and from mother to child during birth. The enduring persistence of hepatitis B in modern populations is not a random accident of recent history. Instead, it is the stark continuation of a 20,000-year pattern of infection, a relentless cycle that has never once been interrupted. What truly makes hepatitis B particularly unsettling is the profound insight ancient DNA analysis has provided into its evolutionary trajectory. The very researchers who sequenced prehistoric hepatitis B discovered a virus that has maintained a remarkable and chilling stability across countless millennia. The lineages that actively circulated in Bronze Age populations are, astonishingly, genetically similar to the lineages circulating today. This virus, it appears, has never needed to rapidly evolve or adapt to remain incredibly successful within human hosts. This ancient pathogen discovered its biological niche within the human liver countless millennia ago, and it has relentlessly occupied that very same space ever since. It passes down through generations with the same unwavering, quiet efficiency it demonstrated in the nascent Neolithic villages where it first took root. The implications of this are vast, reaching into every corner of modern medicine, because hepatitis B possesses an insidious ability to remain dormant for decades, completely undetected, before any symptoms emerge. This means untold numbers of infected individuals unwittingly carry the virus, unknowingly transmitting it to their sexual partners, passing it to their children, or infecting anyone who comes into contact with their blood. The virus cunningly exploits the extended human lifespan, slowly, patiently inflicting damage upon the liver over decades, only becoming clinically visible when the destruction is already severe. In this chilling sense, hepatitis B is perfectly adapted for species with long lives, and humans provide precisely the kind of prolonged host lifespan the virus requires to maximize its opportunities for transmission. But what is truly disturbing about hepatitis B is the immense scale of its prehistoric dominion. Genetic analysis of ancient samples reveals that this virus had already conquered multiple continents by the Bronze Age, meaning it had been a silent companion on human migrations for thousands of years, even before that era. Every surge of human expansion across Europe, throughout Asia, and even into the Americas unwittingly carried the virus along. When our ancestors dared to cross the Bering Land Bridge into the New World, hepatitis B was right there with them. When Bronze Age traders forged pathways across Eurasia, the virus journeyed alongside them. The global map of hepatitis B distribution is not a modern phenomenon. It is, in fact, a viral blueprint of prehistoric human migration, deeply etched into our very DNA, meticulously preserved across 20,000 years of uninterrupted transmission. Hepatitis B continues to plague us today precisely because it has always been here. It predates agriculture, it is older than writing, it is more ancient than the oldest civilizations humanity can even remember. It rode with our ancestors from the Neolithic into every subsequent age of human history, and it continues its relentless journey with us now, silently infecting hundreds of millions across every continent, claiming nearly 1 million lives each year, a testament to its patience and persistence, qualities only an ancient virus can possess. We did not inherit hepatitis B from the contemporary world. In fact, the modern world inherited it from us. It has been carried forward through 20,000 years of ceaseless human transmission, indelibly embedded in the genetic memory of a species that has never truly known a world without it. Now, let's turn our attention to number 5, the Herpes Simplex Virus, a 6 million year companion. Long before humans emerged as a distinct species, before our ancestors learned to walk upright, even before the genus Homo diverged from the broader primate lineage, a virus had already made its home within the bodies of our evolutionary predecessors. Herpes Simplex Virus Type 1, or HSV1, has been co-evolving with hominids for at least 6 million years, a fact firmly established by phylogenetic analysis published in the journal Virology in 2017. Researchers meticulously tracing the molecular clock of the virus determined that HSV1 splintered into its current form around the precise moment our lineage diverged from the ancestors of chimpanzees. The virus, it turns out, was present at the very dawn of humanity's existence, and it has been with us at every single step along the way ever since. Currently, HSV1 infects an astonishing 3.7 billion people worldwide, according to the World Health Organization. That staggering figure represents roughly 2/3 of the global population under the age of 50. This cunning virus establishes a permanent residence within the trigeminal ganglion, a cluster of nerve cells nestled near the base of the skull, where it patiently lies dormant for the entire lifespan of its infected host. Crucially, it cannot be cured. It cannot be eradicated. Once HSV-1 infiltrates a human body, it remains there until that body breathes its last, only reactivating periodically to produce those familiar cold sores, or, in rare and terrifying cases, spreading to other vital tissues, including the brain, where it can trigger fatal encephalitis. What makes HSV-1 particularly unsettling is the profound and intricate relationship it has forged with the human nervous system across 6 million years of relentless co-evolution. The virus doesn't simply conceal itself within nerve cells, passively waiting. Rather than merely hiding, it deeply embeds itself within the very neurological infrastructure of its host. This insidious intruder actively manipulates gene expression within the neurons it occupies, subtly altering the behavior of the immune cells that patrol the nervous system and even responding to delicate stress signals generated by your own body. When you experience physical strain, emotional trauma, exposure to sunlight, or any form of illness, HSV-1 detects these signals, awakening from its slumber. It then travels down your nerve fibers, culminating in a fresh, often painful outbreak. This virus has devoted 6 million years to mastering the profound chemistry of human distress, and it leverages that intimate knowledge to relentlessly maximize its own transmission opportunities. The far-reaching implications of this viral presence extend into areas of research that most people have never even considered. Studies published over the last decade, including pivotal work from both the University of Manchester and the Ruhr University Bochum, have meticulously linked HSV-1 infection to the devastating development of Alzheimer's disease in individuals with a specific genetic susceptibility. Researchers meticulously examining the brains of Alzheimer's patients found HSV-1 DNA precisely concentrated in the very regions where amyloid plaques, the hallmarks of the disease, form, strongly suggesting that the virus may be an active contributor to this pervasive neurodegenerative condition. The profound cognitive decline that tragically robs millions of elderly people of their memories and their essence each year may be driven, at least in part, by a virus that has been a silent, constant companion to our species since before we even became what we recognize as human. The truly unsettling aspect of HSV-1 lies in the sheer intimacy of its presence. This virus is relentlessly passed from parent to child, from lover to lover, an unbroken chain of infection stretching across 6 million years, never once severed across countless generations. Every single human ancestor who managed to survive long enough to reproduce inevitably carried this virus forward, ensuring its lineage persisted. Your grandmother carried it, her grandmother carried it. The very first members of Homo sapiens carried it, and even the Australopithecines before them had it. Every single link in the vast chain of your biological lineage, reaching back to a time long before humans ever walked the earth, has been profoundly shaped by the continuous, inescapable presence of this virus. You are not merely a host that HSV-1 has opportunistically colonized. You are a body that meticulously evolved within a world where HSV-1 had already been patiently waiting for millions of years. Now let's explore our fourth point, endogenous retroviruses and the viral inheritance. It's a startling fact. Approximately 8% of the human genome is not, in fact, human. It is viral. Endogenous retroviruses, or ERVs, are striking fragments of ancient viral DNA that successfully infected our ancestors millions of years ago, becoming permanently integrated into the very genetic material passed down from parent to child. The groundbreaking Human Genome Project, completed in 2003, revealed the true, astonishing scale of this genetic integration for the very first time. Roughly 100,000 distinct viral DNA sequences are deeply embedded within the human genome, representing dozens of different ancient retroviruses that successfully infiltrated our evolutionary ancestors over the last 100 million years of mammalian history. Every single cell in your body meticulously carries these sequences, and with every division of every cell, they are faithfully copied. Genetically speaking, you are approximately 8% ancient virus. The mechanism that brought about this profound integration is disturbing in its sheer simplicity. Retroviruses, the very family that includes modern HIV, reproduce by forcefully inserting their own genetic material directly into the DNA of the cells they infect. When a retrovirus happens to infect a sperm or egg cell, and the host then survives to reproduce, that viral DNA is subsequently passed to every single cell in the body of the resulting offspring. If that offspring then reproduces, the viral DNA is passed to the next generation, perpetuating this viral legacy. Over millions of years, these germline infections relentlessly accumulated. Each successful insertion became an immutable, permanent feature of the human genetic inheritance, relentlessly carried forward by every descendant of the infected individual. What makes endogenous retroviruses particularly disturbing is the critical fact that many of them are far from silent. Research published over the last decade has starkly demonstrated that ERV sequences are, in fact, actively transcribed within human cells, producing proteins that dramatically influence gene expression, immune function, and even fundamental embryonic development. Some of these ancient viral sequences, surprisingly, have even been co-opted by human biology for exquisitely essential functions. Consider the syncytin genes, born from an ancient retrovirus that invaded our mammalian ancestors some 25 million years ago. These genes are absolutely essential for a critical process, the formation of the placenta during pregnancy. Picture this. Every single human being ever born has developed within a placenta partly constructed from these ancient viral proteins. Without that primordial infection, mammalian reproduction, as we understand it, simply would not exist. But the story doesn't end in our distant past. Its implications reach unsettlingly into modern disease. Over the last decade, studies published in prestigious journals like Nature and Cell have uncovered a startling link. The reactivation of dormant endogenous retroviruses is tied directly to the onset of multiple sclerosis, amyotrophic lateral sclerosis, ALS, various cancers, and a host of autoimmune disorders. The viral sequences woven into our very human DNA are far from inactive. Under specific conditions, be it inflammation, acute stress, or the slow march of aging, these ancient viral remnants can partially reawaken. They begin producing proteins that actively damage the very cells harboring them. This means that the diseases claiming millions of human lives each year might, in part, be driven by viral infections that plagued our ancestors tens of millions of years in the past. The truly horrifying revelation of endogenous retroviruses lies in what they expose about the core of human identity. You are not a pure, unbroken biological lineage. You are, in essence, a chimera, a genetic patchwork. Your being is a tapestry of mammalian genetic material intricately stitched together with the viral DNA that infected ancestors long dead. Ancestors whose names you'll never know, whose faces you could never recognize. The traits you instinctively consider fundamentally human, the very structure of your placenta, the delicate regulation of your immune system, the complex architecture of your brain, all have been deliberately shaped by infections that occurred aeons before humanity even walked the earth. When you scrutinize your own genome at the molecular level, you are not gazing upon a pristine human blueprint. Instead, you are peering into a palimpsest, a document written over and over again for millions of years, not by human hands, but by viruses. These viruses are no longer alive, yet their indelible genetic signatures remain deeply embedded within every single cell of every person who will ever live. The viruses perished, but their DNA did not. Instead, it became you. This leads us to a new, unsettling chapter. Number 3. Pithovirus Sibericum and the Siberian Revival. In 2014, a team of audacious French researchers, under the leadership of microbiologist Jean-Michel Claverie, achieved something most outside the scientific community barely conceive as possible. They brought back to life a virus that had been locked in the grip of Siberian permafrost for 30,000 years. And not only did they revive it, but they proved it was still infectious. This virus, christened Pithovirus Sibericum, was painstakingly extracted from a core sample of permafrost taken from a remote region of Siberia known as Chukotka, at a depth corresponding to deposits from the late Pleistocene era. When these researchers introduced amoebas to the thawed sample, the virus, against all odds, sprang back to life. It infected the amoebas. It began to reproduce. After an unfathomable 30 millennia of dormancy, it was still a fully functional biological agent. Pithovirus Sibericum is no ordinary microbe. It's a giant virus, substantial enough to be seen under a standard light microscope, boasting a particle size of roughly 1.5 micrometers. This makes it larger than many bacteria. Its genome contains approximately 500 genes, a staggering number compared to the mere 10 genes typically carried by common human viruses like influenza. This virus belongs to a family of giant viruses, entities completely unknown to science until the early 2000s, whose very existence shatters traditional definitions of what a virus actually is. These are not simple genetic packages. They are intricate biological entities capable of encoding their own metabolic machinery, their own sophisticated genetic repair systems, and their own devious mechanisms for hijacking their host cells. What makes the revival of Pithovirus cybericum particularly unnerving, though, is the stark truth it reveals about the permafrost itself. The Arctic permafrost sprawls across an immense 15 million square kilometers of the Earth's surface, blanketing vast stretches of Russia, Canada, Alaska, and northern Scandinavia. Within its frozen depths lies the biological material of every organism that has ever perished in these regions over the past million years. This includes bacteria, viruses, fungi, parasites, and the tissues of every mammal, every bird, and every human who has ever lived and died upon that ground. All of it is preserved, held in a perfect, chilling stasis by temperatures that have remained relentlessly below freezing since the last ice age ended. The successful revival of Pithovirus cybericum wasn't just a hypothetical exercise. It was undeniable experimental proof that complex viruses can endure 30,000 years of freezing and, astonishingly, remain fully functional once thawed. The implications of this breakthrough ripple across every climate projection for the coming century. The Arctic, a region critical to global climate stability, is now warming at a rate 4 times faster than the rest of the planet. A chilling fact confirmed by research in the journal Communications, Earth and Environment in 2022. This accelerated warming is melting permafrost that has been solidly frozen for hundreds of thousands of years. Imagine, these are regions that have been biologically sealed since before humanity migrated out of Africa, now exposed to our modern atmosphere for the very first time. Whatever ancient viruses lie locked within that ground are now being unleashed into ecosystems that possess no evolutionary memory of their existence. The organisms that might once have served as their natural hosts are long gone, extinct for millennia. The immune systems of modern species have never encountered these relics of the deep past. What unfolds next is a question no epidemiologist can confidently answer. What makes the Pithovirus cybericum discovery truly unsettling is that the researchers weren't even searching for dangerous pathogens. Their goal was fundamental, to test if ancient viruses could survive prolonged freezing. The virus they stumbled upon happened only to infect amoebas, posing no direct threat to humans or other animals. Yet the profound scientific principle established by their experiment reaches far beyond amoeba-infecting viruses. If a 30,000-year-old virus can be reawakened from Siberian permafrost, then every other virus trapped in that frozen ground holds the potential for revival. Pithovirus cybericum was the proof of concept, a stark demonstration that the Arctic isn't merely a graveyard, it's a vast library, and that library is now eerily opening its doors. Then came Mollivirus cybericum, the astonishing second revival. Just 1 year after unearthing Pithovirus cybericum in 2015, the very same research team announced they had successfully revived a second ancient virus from the identical Siberian permafrost sample. Mollivirus cybericum was genetically distinct from Pithovirus, representing an entirely different lineage of giant virus. Yet it had suffered the same 30,000 years, frozen in the same sediment, enduring the same 30 millennia of dormancy. And when exposed to amoebas in the controlled environment of the lab, it mirrored Pithovirus's capacity for reactivation and reproduction. 2 distinct ancient viruses awakened from the same core sample within the span of a mere year. The discovery of Mollivirus cybericum completely reshaped the interpretation of what Pithovirus had first suggested. Pithovirus alone might have been dismissed as a fascinating anomaly, an unusually resilient viral lineage that somehow survived conditions lethal to most viruses. But Mollivirus proved that the survival of ancient viruses isn't a rare occurrence. It's an inherent property of the permafrost environment itself. If 2 unrelated giant viruses could be revived from the same small sample of frozen ground, then the concentration of viable ancient viruses within Arctic permafrost is likely magnitudes higher than any previous estimate. Every cubic meter of thawing permafrost could potentially contain dozens, even hundreds of unique viruses, each one potentially capable of reactivation under the right conditions. What makes Mollivirus cybericum particularly disquieting is its sheer structural complexity. This virus, roughly 600 nanometers in diameter, carries a genome of approximately 650,000 base pairs, encoding over 500 proteins. Its replication cycle necessitates taking over the host cell's nucleus, effectively seizing control of the most protected compartment of its target cell and rerouting its operations entirely toward viral production. The sophistication of this mechanism points to a virus that has been honing its strategies across vast evolutionary timescales, perfecting its ability to hijack cellular machinery that has existed in roughly its current form for hundreds of millions of years. The host cells it infects today aren't identical to those it targeted 30,000 years ago, but they are similar enough that the virus still functions flawlessly against them. The implications here plunge us into a realm of biology that science has barely scratched the surface of. Most definitions don't even consider viruses alive, yet they are the most abundant biological entities on Earth, outnumbering every other life form by orders of magnitude. Researchers estimate that the Arctic permafrost alone harbors more viral particles than there are stars in the entire observable universe. And the chilling truth is this: the vast majority of these viruses have never been catalogued, never studied, never even identified as belonging to any known family. Take Mollivirus cybericum, for instance. It emerged from a lineage utterly unknown to science before its discovery. This paints a stark picture. Whatever else lies frozen in the Arctic ground is almost certainly just as unknown, just as alien to the modern biosphere it will eventually encounter. The truly horrifying aspect of the Mollivirus cybericum discovery lies in the mathematical trajectory it implies. In 2014, one ancient virus was revived. Just 1 year later, in 2015, a second ancient virus was brought back. The research that unearthed these discoveries has continued relentlessly, and the pace of Arctic thaw has only accelerated with each passing year since. The permafrost is now relinquishing its viral material at a rate that completely overwhelms the scientific community's capacity to even begin studying it. Every summer, more ancient ground is laid bare, and each autumn, a portion of what was exposed gets washed into rivers and oceans, flowing into ecosystems that possess no prior experience, no natural defenses against whatever archaic biology it carries. The revival experiments, once meticulously conducted in sealed laboratories, are now being replicated, utterly uncontrolled, across 15 million square kilometers of Arctic territory. The laboratory, it turns out, was never the problem. The laboratory was the urgent and terrifying warning. Let's look at number 1, Pandoravirus yodoma, and the oldest revival to date. In the year 2022, the research team, spearheaded by Jean-Michel Claverie, announced they had achieved the revival of the oldest virus ever brought back to life. This particular virus, christened Pandoravirus yodoma, had been carefully extracted from permafrost sediments, dated a staggering 48,500 years old. It had been frozen since a time when Neanderthals still roamed the Earth, when the colossal megafauna of the last ice age still thundered across the northern hemisphere, and when anatomically modern humans had only just begun their tentative spread across the vast expanses of Europe and Asia. The virus had remained perfectly viable across that entire immense span of prehistoric time. And when exposed to amoebas in a carefully controlled laboratory environment, it infected them with shocking, effortless success. This 48,500-year dormancy record currently stands as the boundary of confirmed viral revival, a boundary that experts agree will almost certainly be shattered. Pandoravirus yodoma belongs to the Pandoravirus family, a lineage of giant viruses first identified in 2013. These are among the largest viruses ever discovered, with individual particles reaching 1 micrometer in size and genomes exceeding 2 million base pairs. Their genetic complexity rivals that of some small bacteria, and the vast majority of the genes they carry possess no known equivalents in any other organism on Earth. Researchers studying Pandoraviruses have stumbled upon proteins they cannot identify, enzymes they cannot categorize, and genetic sequences that appear in no other known life form. These are biological systems that evolved along pathways so utterly different from the familiar branches of life that they might represent an entirely separate domain of biology, one that has been carefully hidden in the permafrost for significantly longer than Homo sapiens has even existed as a species. What makes Pandoravirus yodoma particularly terrifying is the sheer weight of what its age represents. At 48,500 years old, this virus predates every single human civilization. It predates the agricultural revolution, the very first permanent settlements, and the domestication of every single animal humans now keep. It predates the awe-inspiring cave paintings of Lascaux and Altamira. It even predates the extinction of the Neanderthals themselves. When this virus became frozen, modern humans had not yet developed the cultural complexity that would eventually give rise to written language, mathematics, or organized religion. The virus had been patiently waiting in the ground for a duration longer than the entire recorded history of human civilization, multiplied by 10, and it sprang back to life in a laboratory after just a single week of standard revival procedures. The horrifying conclusions drawn from the survival of this one virus echo across every single projection of climate change's impact on human health. The very ground from which the Pandoravirus was pulled, the Yodoma permafrost, stands as one of Earth's oldest and most carbon-rich frozen vaults. It blankets an immense area, roughly 1 million square kilometers, stretching across Siberia and Alaska. This ancient permafrost has held fast, frozen solid, for hundreds of thousands of years, perfectly preserving biological material from an era when our planet's climate, its intricate ecosystems, and the very composition of its species were profoundly different from anything we know today. Current warming forecasts paint a stark picture. The vast majority of this Yodoma permafrost is slated to thaw completely within the next century. This means that countless viruses, far predating Pandoravirus Yodoma, will inevitably be unleashed into our modern biosphere at a pace that utterly overwhelms any current monitoring system. Of course, some of these ancient viruses will only be able to infect amoebas and other microscopic organisms, posing no direct threat to humans. But what about the others? That's the chilling uncertainty. The truly unsettling revelation brought by Pandoravirus Yodoma isn't just its existence, but the terrifying precedent it sets. Before 2014, scientists had never successfully revived an ancient virus. Yet by 2022, that record had been pushed to 48,500 years. The trajectory of this research is undeniably clear, moving in a single, unwavering direction. With each passing year, scientists deliberately target older and older viruses for revival. Each year, our technical prowess to extract and reawaken frozen viral material grows more sophisticated. But even more critically, every year, more permafrost thaws on its own, without any human intervention, spilling its ancient contents, viruses and all, directly into our rivers, saturating our soil, and permeating our very atmosphere. The viruses we revive in controlled laboratory settings represent merely the sanctioned edge of a phenomenon that is already unfolding unchecked across the entire Arctic. The oldest virus currently confirmed to be viable is 48,500 years old. However, the oldest virus actually lying dormant and still viable within the permafrost is almost certainly exponentially older, waiting for its moment. These frozen permafrost regions harbor the remains of humans who succumbed to diseases that have vanished from our modern world. They contain the bodies of animals that went extinct thousands of years ago, along with the very viruses that once ravaged them. They encase entire microbial communities from environments that ceased to exist on Earth for entire geological epochs. As these regions surrender their icy grip, every single element of that ancient biology is being released. While it's true that the vast majority of it will likely pose no threat to modern life, some of it definitely will. The sheer mathematics of probability, stretched across 15 million square kilometers of thawing ground, simply doesn't allow for any other conclusion. Pandoravirus Yodoma was not a mere scientific discovery. It was a stark and inescapable warning. That warning has now been issued, yet the ground continues to melt, and nothing that has been released from the ancient ice can ever be put back.